So today we are discussing about histology of nervous tissue. Under histology of nervous tissue, we will discuss about 4 slides. One is peripheral nerve, longitudinal section and its transverse section, and dorsal root ganglion, otherwise called as sensory ganglion, autonomic ganglion, otherwise called as motor ganglion, dorsal ganglion or sensory ganglion, and autonomic ganglion, otherwise called as motor ganglion. Motor ganglion. Okay, coming to the peripheral nerve structure. Peripheral nerve structure, longitudinal section. Okay, here in the longitudinal sections of peripheral nerve, we will able to see axons. So these are the axons. Usually, when if we want to see the myelin sheath, usually by using eosin hematoxylin staining, we can't able to see the myelin sheath. And the myelin sheath can be seen only if the slide is stained by osmic acid. So here, that's why I have represented the myelin sheath here with black color, because the myelin sheath can be seen if the stain is by staining techniques used osmic acid. If the osmic acid is used, then only we will be able to see the myelin sheath. So these are nothing but the nerve fibers, nothing but the axons, and here we have myelin sheath is present, because we are surrounding the axon, we have myelin sheath is present. Outside the myelin sheath, we have neurofibril neurilemma is present. Outside the myelin sheath, we have neurilemma sheath is present, which is made up of squamous cells. So, but we can't see the neurilemma layer. We can't see the neurilemma layer. Why? Because because we have used the technique, osmic acid, that's why only myelin sheath is present, and this myelin sheath, it is produced by the squamous cells. The squamous cells are present in the neurilemma layer, and this neurilemma layer consists of squamous cells. They produce the myelin sheath. If the axons are myelinated, then the impulse is transferred. Then the transfer of impulse is faster. Okay, when the axon is myelinated, in the myelinated axon, the transfer of impulse is faster. Why? Because here, see here, the myelin sheath, it is interrupted at certain levels. Like, see, the myelin sheath, it is interrupted at certain levels. Okay, these interrupted levels, we call it as, these interruptions we call it as rods of Ranvier. Here what happens is the neurilemma sheath, which is present outside the myelin sheath, it comes in direct contact with the axon. So what happens is the impulse jumps from here, from the one node of Ranvier to other nodes of Ranvier, and one other node of Ranvier to other nodes of Ranvier. So the impulse is going faster. So that is why the impulse is faster in myelinated nerve sheath, because the impulse jumps from one nodes of Ranvier to other nodes. So what are nodes of Ranvier is nothing but the myelin sheath is interrupted at certain levels. They are called it as nodes of Ranvier. Outside the myelin sheath, we have squamous cells are present. These squamous cells, they are nothing but the neurilemma layer is present, made up of squamous cells. These squamous cells are responsible for the formation of myelin sheath. Myelin sheath is made up of phospholipids. Inside the axon, we have neurofibrils are present. Inside the axon, we have cytoplasm is present, that is called axoplasm. And from the axoplasm, along with the axoplasm, we have neurofibrils are also present. These neurofibrils, they will give support to the axon. These neurofibrils are nothing but the thin nerves, thin peripheral fibrils, which are arranged linearly along the axon, and they give support to the nerve fiber. Okay, this is the peripheral nerve longitudinal section. Okay, so in the peripheral nerve longitudinal section, when it is stained by osmic acid, we will able to see the myelin sheath. The peripheral nerve, the axon, it is covered by myelin sheath, and the myelin sheath, it is not continuous and it is disrupted at certain levels. That's why we call them as nodes of Ranvier. Because of the nodes of Ranvier, the impulse goes faster. The impulse jumps from one node of Ranvier to other node of Ranvier. So the impulse is faster in myelinated nerve sheath. This myelinated nerve sheath consists of phospholipids. By using hematoxylin and eosin staining, we can't see the myelin sheath. And outside the myelin sheath, we have one layer that is called neurolemma layer, but we can't see the neurolemma layer by staining with the osmic acid. And this neurolemma layer consists of squamous cells which produce myelin sheath. Okay, so at the nodes of Ranvier, the outside layer we call it as neurolemma layer. We can't see here. It comes in contact, directly in contact with the axon, and the axon consists of neurofibrils as well as the axoplasm. So this is the peripheral nerve longitudinal section. And coming to the peripheral nerve transverse section. This is the transverse section of the peripheral nerve. Here we can't see the myelin sheath. Here we can't see the myelin sheath because we have used it in hematoxylin staining. That's why we can't see the myelin sheath. This is the axon. This is the axon, right? So this is the axon. So the axon here, it is surrounded by a layer, connective tissue layer. That layer we call it as endoneurium. Usually the nerve trunk, it is covered by a connective tissue that is called epineurium, and this epineurium sends septa which divides the nerve into more number of fascicles which nerves the nerve into more number of fascicles, and the epineurium surrounds the entire nerve fiber, and the perineurium surrounds the group of nerve fibers, and endoneurium surrounds each endoneurium. Okay, so the epineurium surrounds the connective tissue, surrounds the entire nerve trunk, and this epineurium connects the sense septa which divides the nerve fibers into groups. And the group of nerve fibers, they are surrounded by perineurium, and each nerve fiber, it is surrounded by endoneurium. So in the staining here, by using hematoxylin and eosin staining, we will see only axons, but usually the axons are surrounded by myelin sheath, but we can't see the myelin sheath here, but we can see the connective tissue surrounding each nerve fiber, that is endoneurium, and the group of nerve fibers, they are surrounded by connective tissue, that is perineurium, and the entire nerve fiber, it is surrounded by a layer of connective tissue, that is epineurium. So that's about the peripheral nerve longitudinal section as well as the transverse section. Okay, and coming to the dorsal root ganglion. Otherwise called as sensory ganglion. Okay, so the dorsal root ganglion, when we take the section of dorsal root ganglion, this is in the dorsal root ganglion in the cell body. When we take the section of the dorsal root ganglion, it consists of prominent nucleus with nucleolus. The cell bodies are very larger. And they consist of prominent nucleus with nucleolus. And the cell bodies, they are surrounded by the multicapsular cells or satellite cells. They are surrounded by a layer of nucleated layer. Their cell bodies are surrounded by a capsule made up of multinucleated layer, and it is otherwise called as satellite cells are present surrounding the cell bodies. And here the cell bodies and nerve fibers, they are arranged in groups. And the neurons present in the dorsal root ganglion is the bipolar pseudo-unipolar neurons. The neurons present in the dorsal root ganglion is the pseudo-unipolar neurons. Because here, this is the cell body and this is the axon. The axon branches like this. Nothing but the one axon is present. The cell body is present in the dorsal root ganglion are the pseudo-unipolar neurons, and these neurons have the efferent nerve fibers. Efferent nerve fibers are present. Okay, these neurons consist of efferent nerve fibers. Okay, the cell bodies consist of efferent nerve fibers, and also the cell bodies consist of prominent nucleus with a nucleolus, and they are surrounded by a multinucleated layer consists of satellite cells. And here the nerve fibers are also arranged in groups. The nerve fibers as well as cell bodies. They are arranged in groups. Here the cell bodies are nothing but the pseudo-unipolar neurons. Pseudo-unipolar cell bodies are seen. Pseudo-unipolar neurons are seen. Okay, these cell bodies have efferent neurons. And here the cell bodies as well as the nerve fibers, they are arranged in groups. So in the dorsal root ganglion, the cell bodies are very larger. Neurons are arranged in groups. See here, the neurons are arranged in groups, and the nerve fibers are also arranged in groups. Surrounding the neurons, we have a multicapsular layer is present. Multinucleated layer capsule, which consists of more number of nuclei present. These cells are nothing but the satellite cells, and the nucleus is present at the center, and the nucleus is also prominent. So this is called epineurium surrounding the entire trunk. Neurons are arranged in groups, and here multinucleated layer present surrounding the neurons, and the axons are also arranged, the nerve fibers are also arranged in groups. So surrounding that we have connective tissue is there, and in the dorsal root ganglion is nothing but it consists of pseudo unipolar cells. They have larger neurons, they have larger cell bodies compared to the autonomic ganglion. Okay, when we take the section of autonomic ganglion, the cell bodies are comparatively smaller. The cell bodies are comparatively smaller, neurons are comparatively smaller when compared with the neurons of dorsal root ganglion. The cell bodies of autonomic ganglion, the cell bodies are comparatively smaller. And here we know that in the dorsal root ganglion, the neurons are arranged in groups and nerve fibers are also arranged in groups. Here, the neurons and nerve fibers, they are not arranged in groups, they are scattered. They are scattered, they are not arranged in groups. See here. Here the neurons are arranged in groups and nerve fibers are arranged in groups. Here, the neurons are comparatively smaller and the nucleus is eccentrically placed with a prominent nucleolus. They are not arranged in group, they are scattered. Okay, and it is consists of pseudo unipolar neurons. It is a multipolar neurons are present. In the autonomic ganglion, multipolar neurons is present. In the dorsal root ganglion, pseudo unipolar cells are present. The cell body has afferent neurons. In dorsal root ganglion, the cell body has efferent nucleon. The cell body has efferent nerve fibers. In the autonomic ganglion, here the nucleus is present at the center, and it is surrounded by a capsular layer consists of satellite cells, and multinuclear condition is seen. Here also cell bodies are surrounded by capsule, but the nuclei are very less. Multinucleated layer is not seen. Here also the cell bodies are covered by capsule, but the multinucleated condition cannot be seen like the dorsal root ganglion. Here the neurons as well as the nerve fibers, they are arranged in groups. Here the neurons and nerve fibers, they are scattered. And then here the nucleus is present at the center with a prominent nucleolus. Here the nucleus is placed eccentrically with a prominent nucleolus. So that's about the dorsal root ganglion and autonomic ganglion. Dorsal root ganglion consists of pseudo unipolar neurons and the autonomic ganglion consists of multipolar neurons. It has cell bodies of afferent neurons. Here the cell bodies consist of efferent neurons. Okay, and here the neurons are arranged in groups, here the neurons are arranged scattered, and the nerve fibers are also scattered. Here nerve fibers are arranged in groups. Okay, so that's about the dorsal root ganglion. Autonomic ganglion, otherwise called as motor ganglion. So that's about the histology of nervous tissue. So we will study about 4 slides. One is peripheral nerve fiber longitudinal section. Here we can, with the stain using osmic acid, we will able to see the myelin sheath here, and by using eosine hematoxylin we can't see the myelin sheath here. Instead of that, we will able to see that endoneurium, nothing but connective tissue layers surrounding the each nerve fiber, as well as the perineurium surrounding the group of nerve fibers, as well as the epineurium surrounding the entire nerve trunk in the transverse section of the peripheral nerve fiber. And here the autonomic ganglion consists of autonomic ganglion consists of motor ganglion, is also called as motor ganglion, and dorsal root ganglion, otherwise called as sensory ganglion. In sensory ganglion, the difference between the two ganglions are represented here. These are neurons are arranged in groups, nerve fibers are also arranged in groups. Here the neurons as well as nerve fibers, they are scattered. The difference between the nucleus centrally placed and here the eccentrically placed. Here consists of pseudo unipolar neurons, here consists of multipolar neurons. Here nerve fibers are afferent and here the nerve fibers are efferent, and also the cell bodies are comparatively larger, and here the cell bodies or neurons, they are comparatively smaller. Okay, and the cell bodies here, they are surrounded by a capsule consists of multinuclei, and here the cell bodies also consist of capsule, but the nuclei are that much prominent and they are not much more present like the nuclei present in the capsule surrounding the neuron present in the dorsal root ganglion. So that's about the difference between the dorsal root ganglion and the autonomic ganglion. So this is the dorsal root ganglion and this is the autonomic ganglion. That's about the histology of the nervous tissue.